Picture a weapon so precise it can shut down your nerves in seconds, melt through muscle like butter, or quietly start digesting you from the inside out, all before you even realize you have been hit. Now imagine this weapon is not a machine, but a living, breathing creature, carrying it in microscopic sacs hidden behind fangs or buried in tiny needle-like mouthparts. Inside, it is not just poison. It is a shimmering molecular orchestra with hundreds or even thousands of different molecules, each playing its part in a deadly symphony. Among nature's most brilliant and ruthless chemists are snakes and spiders. For millions of years, they have fine-tuned their venoms into tools of breathtaking complexity. These formulas are so advanced that even our most sophisticated labs struggle to reverse engineer them. These brews can freeze prey in place, liquefy organs, or make a predator regret its life choices in an instant. The deeper scientists peer into this microscopic world, the clearer it becomes. These venoms are not just dangerous, they are living masterpieces of evolution. Welcome back to the Curious Playbook, where we explore the hidden science behind everyday wonders and the not-so-everyday ones that could, in theory, stop you in your tracks. Today we are diving into a fascinating question. Between snakes and spiders, whose venom is more complex? By the end, you will see why this battle is not as simple as counting toxins, and why both contenders hold secrets that could save lives or end them. Venom systems themselves are astonishing proof of natural selection in action. They did not evolve just once. They have appeared again and again in unrelated groups such as jellyfish, cone snails, scorpions, fish, and even the platypus. Every time, evolution found a way to turn basic biochemistry into a survival strategy. The goal is always the same: disable prey quickly and deter anything that might want to eat you. What is remarkable is how differently that goal can be achieved. Some venoms attack the nervous system, others the blood, others the muscles, and many do a bit of everything. Each formula is shaped by the predator's diet, habitat, and the threats it faces. To understand this complexity, scientists use venomics, a suite of tools that sound like they belong in a science fiction lab. Proteomics maps every protein in a venom sample down to low-abundance molecules that older methods would have missed. Transcriptomics shows which genes in the venom gland are switched on and making toxins. Genomics reveals how many toxin gene copies an animal carries, which can influence how potent or varied its venom is. With tools like AlphaFold 2, researchers can predict the three-dimensional shapes of toxins. This helps them see exactly how a venom molecule latches onto a nerve receptor or breaks apart a cell membrane. Put it all together and you get systems venomics, a panoramic view of venom from gene to effect. Snake venom has been studied the longest, and for good reason. Its effects on humans are dramatic and often deadly. Over 95% of its dry weight is proteins and peptides. The heavy hitters include phospholipase A2, an enzyme that slices into cell membranes and causes anything from muscle destruction to nerve disruption. Snake venom metalloproteinases are brutal on connective tissue, breaking apart collagen and laminin to cause hemorrhage and swelling. Snake venom serine proteinases hijack the blood clotting process, sometimes causing dangerous clots, other times thinning the blood until it cannot clot at all. Then there are the three-finger toxins, dominant in many elapids like cobras and kraits, which bind to nerve receptors and block the signals that tell muscles to contract. The result is paralysis. Supporting players like hyaluronidase act as spreading agents, letting other toxins move faster through the victim's body. Some venoms carry beta-bungarotoxin, which prevents nerve cells from releasing the neurotransmitters that trigger muscle movement. Others include L-amino acid oxidase, which damages cells and disrupts blood function, and even acetylcholinesterase to break down nerve signaling chemicals. What makes snake venom even trickier is that it is not static. The venom from one species can differ wildly from another, even if they are closely related. Geography matters. The same species in different regions may have different recipes. Age matters. Juveniles and adults often produce different toxin profiles to match their prey. Diet matters. A sea snake that specializes in eel-like fish might have venom dominated by neurotoxins, while one with a broader diet might balance neurotoxins with tissue-damaging enzymes. Even sex can influence venom composition, though individual variation often overshadows it. Spider venom, in contrast, long carried the reputation of being mostly about small neurotoxic peptides. These inhibitory cysteine knot molecules are incredibly stable and deadly to insects, making them perfect for quick immobilization. But modern venomics has revealed a staggering hidden diversity. There can be hundreds to thousands of components in a single species venom, including more than 140 different enzyme families across 17 spider families. Some of these enzymes are directly toxic, like phospholipase D in brown recluse spiders and their kin, which causes tissue death and sometimes systemic illness. Others, like acetylcholinesterase, disrupt nerve signaling in ways similar to some snake toxins. There are predigestive enzymes that liquefy tissues before the spider starts eating, spreading factor enzymes to help other toxins circulate, and even preservative enzymes to keep venom potent inside the glands. In some species, enzymes make up nearly half the venom. That is as much as the neurotoxins themselves. This suggests a two-stage strategy. First, a broad enzymatic attack to weaken prey, followed by a precise neurotoxic strike. Spiders also have a wild card. Many spider venom glands host bacterial communities that do not just live there. They may contribute their own bioactive molecules to the venom mix. Some of these bacterial products have antimicrobial effects, which could help protect the spider's venom glands from infection. Others have shown anti-inflammatory or anti-tumor activity in lab tests. This opens the door to the idea that spider venom complexity is not just about the spider's own molecules, but about a co-evolved partnership with its microbiome. This is something only lightly documented in snakes so far. Both snakes and spiders show remarkable venom variation driven by ecology. A broad diet often means more complex venom to handle different types of prey. A narrow specialized diet can lead to more concentrated venom targeting a specific prey type. Environmental pressures, from climate to prey behavior, shape the recipe. Genetic regulation adds another layer. The number of toxin gene copies and the way non-coding RNAs control their expression can alter venom potency and composition. The question of which is more complex depends on how you measure complexity. Snake venoms tend to be dominated by a few major toxin families, but each is multifunctional and often synergistic with others. Spider venoms have a broader spread of toxin types, especially enzymes and potentially an extramicrobial dimension. Snake venom research is deep and detailed, making it seem simpler because we have already mapped much of it. Spider venom is newer territory, and every year reveals more toxinological dark matter. Venoms also come wrapped in myths. Snakes have been accused of hypnotizing prey, which is more likely just the prey freezing in fear. Ancient stories claimed vipers would bite themselves to avoid capture, which is false. Spiders have their legends too. The tarantella dance of Italy supposedly cured tarantula bites, though the spiders were harmless. The daddy longlegs myth claims they have the most potent venom but cannot bite humans. This is pure fiction. Beyond danger, both venoms are treasure troves for medicine. Snake venom has given us drugs for high blood pressure, heart failure, and blood clots. Spider venom is being investigated for non-addictive painkillers, stroke treatments, and epilepsy therapies. The same specificity that makes these toxins deadly to prey makes them valuable to scientists looking to target a single receptor or enzyme in the human body. So which wins the complexity crown? If we judge by sheer diversity and novelty, spider venom is currently pulling ahead. This is not because it is deadlier to humans, but because it contains an enormous range of enzymes, plus the intriguing contribution of symbiotic microbes. Snake venom remains unmatched in the depth of our knowledge and in its perfected use of a smaller set of devastating molecular tools. The truth is, both are champions in their own right, shaped by millions of years of evolutionary arms races. Every new study suggests we have only scratched the surface. Whether it is the coiled viper in the grass or the patient spider on its web, each is carrying a portable, highly tuned chemical arsenal. They remind us that some of the most sophisticated weapons on Earth are carried by creatures you could step over without noticing. The line between a deadly molecule and a life-saving drug can be as thin as a strand of silk. So the next time you see a snake or a spider, do not go leaning in for a closer look and whispering about how amazing their venom is. That is a quick way to end up on the wrong end of a biology lesson. Appreciate them from a safe distance, preferably one that involves you on the other side of a sturdy window, or better yet, watching a documentary in your pajamas. Let the scientists do the handling, and let us do the curious gawking. If you enjoyed this dive into the microscopic arms race between snakes and spiders, hit that like button, subscribe, and turn on the bell so you never miss our next adventure into the wild, weird, and occasionally dangerous side of science. Trust me, the next topic might make you even more grateful for a nice venom-free cup of tea.